Director of the National Key Laboratory of Crop Genetic Improvement, Professor Qifa Zhang.
Okay, thank you again. Actually, this was picked by Apichat as from the Apichat to ask my student to come to give a talk, but he didn't come. So it's a miscommunication. I use my version to give a presentation. Yet, everybody here knows there are two major rice groups. Actually, that have been recognized in China for very long. So, two rice groups recognized more than 2,000 years ago in Han Dynasty. One as Xian, the other as Ken. The Xian group is now called Indica. The Ken group now called Japonica. And these two groups have very distinct geographical distributions and a number of characteristics. So genome diversity uncovered by whole genome sequencing showed that these two groups are very well separated. So Indica group and Japonica group with very distinct morphology all the way to our eating. Yet if you make a hybrid between these two groups, typically this is what you are going to see. You will see high heterosis, and then you will see low fertility. And then that has generated great interest for overcoming the fertility problem to make use of heterosis of these two groups. So in the past, there are many studies. Genetic analysis identified 50 plus loci for Indica Japonica hybrid sterility. There is a third group called wide compatibility. So this group has the ability to produce normal fertile hybrid when crossed to both Indica and Japonica varieties. So this is basically what it does among these three groups. If we make a cross between typical Indica and Japonica, that is the one at the very bottom, we are going to get sterile plants. Then if we make cross between Indica and the wide compatibility group, and then we see normal fertility. And the other way around, if we make a cross between Japonica and the wide compatibility group, we'll also see normal fertility. Then Professor Ikahashi in Japan, he designed a sort of cross called three-way cross to do genetic analysis for analyzing so-called wide compatibility genes. So that is something like this. Make a cross between Indica and wide compatibility, get F1, and then cross to a Japonica variety. This is what you are getting. And then we could see the segregation of the population into something like this. And this data can be used for genetic mapping or genetic analysis to infer number of genes. It can be mapped to linkage groups. This is what we did in the 1990s, 20 years ago. So we mapped, we get something like QTLs for several loci governing Indica Japonica hybrid sterility. So one of them I would like to mention in particular is S5, which is the most important so far identified. So we also showed the S5 locus is very specific. It causes embryo sac sterility in Indica Japonica hybrid, and that is where the S5 is. Then we developed neoisogenic lines by introgressing this fragment from different varieties into different backgrounds. So in Nanjing 11, which is a typical Indica, by introgressing ones from Japonica, ones from wide compatibility into Nanjing 11 genetic background. Also ones into Balila background, which is a typical Japonica variety. So a number of crosses and back crosses, so we get neoisogenic lines. And finally, we're able to map the sort of behavior into a chromosome segment, which is this long, 40 kb, containing 5 open reading frames. Then we tested, another one. So at the same time, there's another group also published their mapping. So the two mappings overlap by 2 open reading frames. As said, in our mapping region, we had 5 open reading frames. In theirs, we also have a longer 6 open reading frames. So the two mappings overlapped by two open reading frames. Then we started to do transformation analysis using, basically that's a strategy using indica to do transformation with japonica. So transform indica genomic fragment into japonica. We transform three of them after 5 open reading frames, OF3, OF4, OF5. And then if by expectation or prediction, if the gene is right, it's a right candidate, this is what we're getting. So we have japonica, which is the endogenous, plus one that is introduced, SI, then that will cause hybrid sterility. Indica japonica hybrid sterility. Then what we're expecting to see is a reduced spiglet fertility and also embryonic fertility, but we'll see normal pollen fertility. That is our expectation. So we did the transformation of the OF3, 4, and 5. So OF3 didn't cause any fertility problem, neither did OF4, but OF5 did. So OF5 reduced greatly the embryo sac fertility of the transgenic plants. And then we also carried this to the next generation. This is what it did. If we look at the panicles, this is a negative, transgenic negative, it's normal, fertile. Transgenic positive, we don't see any seed setting. So it was greatly reduced hybrid, reduced fertility. So it caused a problem with the embryo sac, not a problem, no problem with the pollen. And then that is the data we obtained for the T1 generation. So T1 generation caused a big problem for spiglet fertility. Also big problem for embryonic fertility, no problem for pollen fertility. So in agreement with our prediction. So only S5, not only OF4, not only OF5, not 3, not 4. So OF5 of the is predicted to encode the protein, like Darwin mentioned, aspartic protease. And that is a structure of the gene. It has a signal peptide and active regions for activities, 2 active sites. So, but it has some sort of structure. If we do some structure comparison, we see the structure features of the 3 classes of the germplasm. So indica, japonica differed by 2 nucleotides, which both of them cause substitutions of amino acid. So one, the wide compatibility variety has a deletion of 150 amino acid, which cause problem with the signal peptide, portion of the N-terminal. And then if we do our sort of subcellular localization analysis by TM localization, and then we'll see typical or normal indica variety of the S5 or the OF5 aspartic protease has a cell wall localization, and so is the typical japonica also has a cell wall localization, not the wide compatibility variety. So it is localized in the cytocell or cytoplasm. So that is some sort of data at one step. So we identified OF5 that could cause embryo sac fertility. But how do these differences, which differed only by 2 amino acids, could cause hybrid sterility? And also by our sort of rationalization, it is difficult to explain the hybrid sterility by this single open reading frame alone. So we did genetic analysis again, and we made a number of crosses to see what is happening if we look at F2. So what happens is segregation distortion in the progeny generation. So if we cross between 2 near-isogenic lines, we immediately see segregation distortion in F2. So very heavily distorted against japonica genotype. make a cross between two normal varieties, Nanjing 11 Balila, we'll see very similar feature, so very heavily distorted against Japonica genotype. And then Nanjing 11 Nipponbari, very similar story, 19311 and Nipponba, we get very similar result. Segregation distortion heavily against the Japonica genotype. Then we rechecked again. We said we have 5 open reading frames. And then we transformed 3 open reading frames. So we did sequences of all the 5 open reading frames. We found structural differences only in 3 of the 5 open reading frames, which is OF3, OF4, OF5. All of them we did the transformation. So those are the features. And then if we put together, those are the summary of the features. So OF5, as we said, we have the indica japonica differed by 2 nucleotides, both of them cause substitutions of amino acid. And then the wide compatibility group with a missing nuclear signal peptide. And then if we go to OF3, which encodes heat shock protein 70, so then it differed at the very end, very terminal, C-terminal, very short fragment. So the C-terminal, the Japonica ones has a short deletion, so is the Dula, which is a wide compatibility variety, and also OF4. OF4 we see very big structural difference between Indica Japonica. Indica variety has a very big deletion at the C-terminal part. The Japonica variety is supposedly we have a full version of it. So we did genetic analysis again. Let's look at the result of the genetic analysis. And then we said if we have a cross between Indica and Japonica, like this one is typical Balila Nanjing 11, we see a reduced fertility. Balila Nanjing 11, so very similar, half fertility, half fertile or half sterile. But if we add OF3, the plus allele, into this cross, look at this. Balila Nanjing 11, half sterile or half fertile, half sterile. And look at Balila Nanjing 11 with OF3, then it becomes 75. So the fertility is greatly increased. What happened? So based on this result, we sort of inferred OF3 plus may have a rescuing effect of IJ hybrid sterility, IJ hybrid fertility. And then OF4, what does OF4 do in this system? So we have Dula Nanjing 11, so it has this sort of genotype, and with OF4, but neither of them has OF4 functional. But if we add OF4 to this cross, then we immediately get greatly reduced fertility. So you take my word for it, OF4 is indispensable for this curing. That means for the reduced fertility. And then we put the rows of these 3 OIFs together in a gamete killing process. OF5, what it does is to initiate the killing process, and then OF4 is a partner of the killing, and OF3 is protecting the gametes from being killed. So these are the function of the 3 genes in this system. So we did subcellular localizations of the open reading frame. We already said OF5 is localized, the normal ones localized in the cell wall, and the mutant localized in the cytosol. So OF3, if we look at the result here, OF3 is localized in ER and Golgi body. OF4 is localized in the membrane. So those are the subcellular localizations. And then based on this result, we propose a model, something like this. So we make a cross between Indica Japonica. This is what we're getting, the hybrid, which is heterodic hybrid for OF3 plus and minus, OF4 plus and minus in the other way around, and OF5 plus and minus. So Then OF5 encoded by this the gene goes to the extracellular space, and then OF4 produced by the genes. The functional one localized on the membrane, non-functional ones doesn't go to the membrane. And OF3, the functional ones localized in ER, and no differences between functional and non-functional. And that is in the stage of megaspore mother cells, and then coming to megaspore formation stage. And then when these two chromosomes or haplotype segregate, so one would get this one. This is an indica genotype. Indica genotype has a functional OF3, functional OF5. That is what is encoded and then localized into the space. At the very end, OF3 will be okay, and then this one become viable. That is a part of the story. What happened is here in the Japonica part. So Japonica genotype carry on a functional OF3 at this stage, which should be still okay, and OF4 functional localized on the membrane domains, and the OF3 has a role rescuing, so by acting or counteracting the function of this one, protecting the gamete, and then eventually this one ran out of OF3 plus and got problem. So, and then eventually became abortive. So that is a genetic model we proposed for the system. And then we did analysis to confirm what happened in this genetic model. Underneath the genetic model, we did differential expressing analysis using chip. Then we saw OF3 get overexpressed, get elevated expressing in OF5 transformant in the microarray analysis. So we saw immediately here OF3 got elevated expressing. And then together with OF3 is a bunch of genes which are ER stress responsive. ER stress responsive. So a bunch of genes with ER stress responsive all get elevated analysis, which the pattern of this is highly similar to a study observed in ER stress study RISE, which was published this year. And then another bunch of genes is PCD-related, Darwin just described. PCD-related genes also got induced in the transgenic, in the OR5 transgenic plants. And then putting this together, we'll also see by cytological analysis, we see the sort of PCD by tunnel assay of embryo sac during megaspore formation stage in Balila in comparison with Balila with transgenic OF5. We see abnormal, so premature PCD of the transgenic OF5 with OF5 transgenic, OF5 plus transgenic plants, and is studied earlier. The signal is much stronger as the development proceeds. And finally, we come up with the biochemical model. So this is some sort of substrate there, and then with the function of OF5, and it produces some sort of product. And product triggers a signal, and then OF4 would respond to the signal, and then cause ER stress. At the presence of a functional 3+, this won't happen. ER stress would not happen. The cell is normal. But when these OF3 is not functional, so the cell is not able to resolve the ER stress, which would cause then some sort of processes which eventually will lead to PCD, and then would eventually cause embryo sac or abortion. So basically, I would like to summarize the result by this sort of analogy of the functions played by these three open reading frame, which is drawn by the student who was supposed to give this talk. So basically, three elements are here. So OF5 is probably an arrow. So the 5+ is a functional arrow. So the 5 minus is a gene is broken. It's only half. It doesn't have the head. Arrow without a head, and the neutral one is an arrow without the other part. So a ball which is played by OF4. So this and this together would cause a killing reaction. So OF3 is a shield. When the shield is functional, it could protect the gamete very well, so the gamete would not die. So when the shield is broken, it is not able to protect the gamete would be killed. So basically, that's what we found in this study. So I would like to acknowledge the contributors. This is a work for more than 20 years. And we made the first cross, I just checked, in 1990. So now it's the year of 2012, so 22 years. And then many students get involved in this work, and also collaborators in other institutions, universities, and also competitors. We sort of resolve the issues, and then we are together to complete the work. And then also acknowledge the financial support by national project, high technology project, National Science Foundation, and also 973. Thank you very much for your attention.
Friends, you will agree that this wonderful work has reached a very great height in exposing biological processes. There is already a question there. Yeah, please.
So very interesting story. I know this work is 25 years story.
22.
21. So question is that the biochemical evidence of the component, 3 components. The S5 is the aspartic proteinase.
Right.
Some literature indicated that aspartic proteinase can anti-PCD, not trigger PCD. Our evidence suggests aspartic proteinase can trigger PCD. Do you have biochemical evidence?
Well, what we propose here is a pathway. So aspartic protease is a trigger. So ER stress, well, the whole process, the genetics, aspartic protease triggered ER stress and then PCD. So probably also some intermediate procedure for process. So this is as much as we know. The other, the two, what we have in the steps, we only have the steps, the arrows, we don't have the detail. So the arrows represent hypothetical steps. We don't know the biochemistry, we don't know the chemicals, we don't know the substance.
So another question is for the explained pattern of the ORF3 or ORF4, specifically in the female organ, the explained pattern.
Express, not necessarily, but OF4, OF3, OF3 is expressed almost everywhere, but highest level in the female developmental ovule, highest level in ovule development, but it's everywhere. So OF5, OF4 expressed at very, very low level, very low level. At the female reproductive stage, it's only slightly higher than background. Otherwise, it's at the background level.
All right. If there are no more questions, thank you very much, Professor Zhang.
All right.
Friends, I am sure that you have enjoyed the scientific content richness of this session.